Okay, so today we're going to be talking about structure and function, mostly cell structure, and how the parts of a cell work together to create this building block of life that can actually be an organism itself. So to first really analyze cells, we need to understand that there are two main categories of cells. And so these are very broad umbrella terms that splits all cells into either prokaryotic or eukaryotic. So the main difference between prokaryotes and eukaryotes is that the prokaryotic cells lack something called membrane-bound organelles. So obviously organelles sounds like organ, and so they're just basically smaller organs that work in the cell. Now, all cells are enclosed by something called a membrane. And you can think of your membrane as like the skin for a cell. It basically encloses it and separates it from the surrounding area, so it keeps all of the cell parts together. And so within this skin membrane enclosed cell, there are more things enclosed with more skin that performs specific tasks. Okay, and these are called organelles. So if you're like, I'm still confused on the membrane, well, there's more on that later. So it doesn't make sense right now. Just remember that eukaryotic cells have organelles, prokaryotic cells don't. So prokaryotic cells are usually bacteria and archaea. And so there are three big groups of life: bacteria, archaea, and eukarya. And the first two groups are prokaryotes, and eukarya has eukaryotic cells. And so eukaryotic cells are bigger, they're more complex, they have organelles. So within the cell, there's like tiny organs, so they have to be bigger. And so we have eukaryotic cells because obviously humans are very complex. We have a lot of different systems in our body, and also plants, fungi, and protists, which are another kind of eukaryote. So even though there's like, oh wait, one more thing. So the meaning behind the word prokaryote, actually, okay, so I will focus on eukaryote first. So the meaning behind the word eukaryote is that eu means true, and kary means like kernel or nut, and so that refers to the nucleus. And so the nucleus is an organelle. So basically, the etymology behind eukaryote is basically just, wait, give me a second. Okay, wait, never mind. Sorry, temporary interruption from parents. Okay, eukaryote just basically means true nut, so like true nucleus, and then prokaryotic is false, no kernel, no nut, yeah. So even though there's like this two main types of cells, there are four common denominators between all cells. So again, there's a plasma membrane, the skin of the cell that encloses it all. There is a cytosol, which is basically all of the liquid inside of the plasma membrane. And there's DNA, or genetic material, that passes down the cell's genes, allowing it to reproduce. And also ribosomes. So ribosomes create proteins. And as you hopefully remember from last class, proteins are very important and play an important role in our bodies. So I think something that things like the visualization on the right kind of skim over it is that different kinds of cells have different numbers of certain kinds of organelles. And so some cells can have more than one nucleus. And like this representation, which is what you usually see, doesn't really showcase that because it just shows a standard cell. So different kinds of cells have different numbers of each of the colored things. And that's what gives it its unique properties. Scienceful questions are throwing shade. We got one today, which which simple sugar is eaten a lot by Americans or something. Oh, I mean, it's not wrong, you know? Yeah. Americans are unhealthy. Well, maybe I shouldn't say that on a recorded Zoom call, but I mean, okay, slight tangent. Scienceful questions always have like some fancy spin that provide like a whole backstory before you actually get to the question. I feel like that's an example of that, but interesting. Thank you for sharing. Anyway, so moving on to the actual common denominators of all cells. Again, first we have the plasma membrane, the skin of the cell. It encloses it. And so I guess what's different from our skin and the plasma membrane is that the plasma membrane is something called semi-permeable. So this means that some substances can pass through it, but only certain substances. So it's selective. When the cell needs certain nutrients, this membrane is going to serve as a door that lets it in. But if it doesn't need nutrients, well, it's just going to not let it in. So thankfully, our skin doesn't like open and close like whenever we see food. That we have other things for that. But in a cell, when they don't have that much control, you use the plasma membrane to regulate the substances that go in and out. So this plasma membrane also deals with the surface area to volume ratio. So surface area is the amount of space that an outer covering covers, while the volume is the amount of space that the covering encloses. So the surface area to volume ratio is what limits cell size. So cells usually can't go past 10 nanometers is what or sorry, 100 nanometers, I think. Yeah. Yes, okay. But there's a reason for that. Otherwise, cells would just keep growing. So if a cell gets really big, then the plasma membrane can't like transport enough nutrients throughout the cell to reach all parts of the cell. So eventually parts of the cell are gonna like die and wither off from not getting enough nutrients close to the plasma membrane. Because obviously the closer you are to your skin, the more like nutrients you're gonna get if it's incoming right away. So basically, just remember in cells, the higher the surface area to volume ratio, or the more surface area you have for like the volume that you have, the more efficient and successful your cell is going to be. And so the plasma membrane, the actual structure part, it's made out of something called a phospholipid bilayer. Again, hopefully you remember what phospholipids are. They are a fat with a phosphate head attached. So the fat part is nonpolar. It doesn't like water. That's why oil and water don't mix. But then the phosphate part, which is where the phospho part comes from, it likes water. So the two lipid tails, so the reason why it's called a bilayer is that it's because it's two phospholipids thick, and the tails face each other while the heads face outwards towards the environment, as you can see in the diagram. And yeah. All right, so second common factor is not the cytoplasm, but the cytosol. The cytoplasm is not found in all cells, but basically inside all plasma membranes, there is a jelly-like substance called the cytosol. And in it, things like DNA and ribosomes are suspended. And so in a eukaryotic cell, the cytoplasm is just basically the cytosol plus organelles. Yeah. Okay. So the main differentiating factor between prokaryotes and eukaryotes would probably be the existence of a nucleus versus no nucleus or nucleoid. So the nucleus is where all of the DNA, the genes, the chromosomes which have genes on them are contained. So I don't know if like your teachers have told you about the cell city model, but oh yeah, it does kind of look like the Earth. Like all of the green is like the continents, that's what they're called, and then the blue is the ocean. But yeah, interesting. Okay, so in prokaryotes, they don't have a nucleus that encloses all of this gene material. In eukaryotes, they do have a nucleus. So even though they don't have a nucleus, prokaryotes still have like this region where all the genes congregate called the nucleoid. And so that's their like rip-off Walmart version of the nucleus. So the nuclear envelope is a double membrane that surrounds the nucleus. So this just means that there's two of the phosphobilayers. And we will see the double membrane appear in other organelles as well. In the nuclear envelope, there are a lot of pores in which materials can pass through, and pore complexes, which are proteins that regulate what can go in and out of the pore. And then the nuclear lamina is a structure that supports the nucleus. So it's structured, and chromatin is DNA plus protein that makes up chromosomes. Again, chromosomes have all of our genes. We have 46 chromosomes that contain all of our genes. And in the nucleolus is something within the nucleus, and this is where ribosomal RNA is synthesized. And this basically gives rise to ribosomes, which smooth transition is the next slide. Okay, so ribosomes are not organelles because they are not covered by a membrane. Instead, they're made out of RNA and proteins. So there are usually two of these subunits in eukaryotes. The subunits are called 40S and 60S, and in prokaryotes, it's 30S and 50S. You don't need to know the subunits at all. I don't know why I put them there, but also, so ribosomes basically help with protein synthesis. So if you remember from last class, DNA makes a copy into the form of RNA, which makes a copy into a protein. And so this RNA to protein step is called translation, and ribosomes perform this function. So there are two main kinds of ribosomes. Only the first kind is found in prokaryotes because, again, they don't have any organelles, and the rough ER is an organelle we'll look at later. So free ribosomes are ribosomes that travel in the cytosol, and usually the proteins that they make are cytoplasmic. So that means that they travel also in the cytosol. Then in eukaryotes, there's something called bound ribosomes, which binds to the rough endoplasmic reticulum, and these proteins are usually destined for things like being inserted in the plasma membrane to help like certain molecules pass through. They're also packaged into lysosomes to help lysosomes perform their function, which we'll look at later. And so, okay, so now that we've looked at all four of the main components that all cells share, we're going to be moving to the eukaryote specific ones. So I guess one of the main systems in the eukaryotic cell, besides the nucleus, of course, is called the endomembrane system. And so this includes a lot of organelles. And they're all classified into the system because they're all either connected directly through a membrane or indirectly through vesicles. So vesicles are basically membrane sacs made out of membrane. So I don't want to make this analogy, but like if someone took skin and made a handbag out of it that can carry stuff from place to place, that would be a vesicle. So the first, I guess, main part of the endomembrane system that is connected to the nucleus, specifically the nuclear envelope, is called the endoplasmic reticulum. And it's this really interwoven network of membranes. We'll see visualization on the next slide. And so there are two types of membranes that stem from the nucleus. So you have your smooth endoplasmic reticulum, and it's called smooth because there's no ribosomes on it. So quickly moving on to the next slide, as you can see, the more coral colored one, it's very smooth. There's no little dots, and those are ribosomes. And so that's why I call it smooth. And so it has many functions. It synthesizes lipids or fats. So it makes steroid hormones, oils, and phospholipids for more membranes. It also detoxifies drugs and poisons. So usually in liver cells, you will find a lot of smooth endoplasmic reticulum that detoxifies, yeah. It also stores calcium ions. So muscles use calcium ions, which are Ca2+, to help move. So every time we raise our arm, the muscle cells in muscles get signified to release this calcium. And so usually you'll find a lot of smooth ER in muscle cells as well. So going back to the idea that different cells can have a different number of things, this is a pretty good example. So liver cells will have more smooth ER than like, I guess, bone marrow cells. And so that's just some differences. So again, all of the diagrams I'm going to be showing, it's very narrow. It's just one representation with everything, but some cells might not even have smooth ER. And yeah, so rough ER is the other kind of endoplasmic reticulum. And if you have not inferred this already, ER stands for endoplasmic reticulum. And so the rough ER has ribosomes studded on the outside, as you saw in the next slide. It looks rough. If you like touched it, it would feel bumpy and not smooth. And so this also has different functions. So the rough ER deals more with proteins. It makes protein or peptide hormones, and it also forms glycoproteins on the membrane. So glycoproteins are proteins with a carbohydrate attached to it. And in the plasma membrane, it deals with signaling and telling the membrane to do this or that. But both of these ER make phospholipids that make up the plasma membrane. Vesicles, we already talked about that. This is a visualization. Okay, so part 2 of the endomembrane system. So let's say you have something that was made in the nucleus, and then it went through the ER. The next stop is the Golgi Apparatus or Golgi Apparatus. And so it's basically like a shipping dock for the products from the ER. And so you saw a lot of flattened membrane sacs in this. It's basically this, but more connected with each other and separate from the nucleus, as you see here. And so there is a distinct directionality. So just like how there's right, left, the Golgi apparatus has something called the cystized the cis side and the trans side. And then in the middle, it's just called the medial side. But yeah, so the cis side receives the materials it's going to send out through vesicles. And the trans side ships out the materials that it receives. And it's on the opposite side, furthest from the ER. And the trans side is also what forms vesicles that moves to other places. And so the Golgi apparatus also forms sugars like pectin and also a bunch of other sugars that is not cellulose found in plant cell walls. So plant cell walls are mostly made out of cellulose, but the parts that are not, a lot of it is formed in the Golgi apparatus. A lot of people also call the Golgi apparatus like the Golgi complex or just Golgi. Yeah. So there is a model that sort of determines or attempts to analyze the behavior of the Golgi apparatus. So the Golgi apparatus, so anytime you find yourself with like a flattened sac that goes back and forth, it's going to be called the cisternae. So the ER had it, the Golgi now has it, and the mitochondria will have it. We'll look at it later. And so basically, the Golgi is constantly moving from the cis side to the trans side. So it like recycles the materials. Okay, so lysosomes are not the next step for all shipping materials that get received to the ER, but it's basically a vesicle with a lot of enzymes that can digest parts of the cell. So going back to last class, enzymes are biological catalysts that speed up chemical reactions. So basically, lysosome enzymes speed up the digestion of certain materials. And so it often undergoes something called phagocytosis, which is the eating of material from a vesicle. And it can combine with a food vacuole to digest all the fuel. Or, yeah, if you don't know what a vacuole is, that is the next slide. Oh, wow. So hang in there. So there is something called apoptosis that all cells are programmed to go through. It's basically programmed cell death. So it's like when cells reach a certain point, they know they can't function as well as a new cell before, but they also don't want to let their material go to waste. So essentially they like digest themselves, and basically the lysosome with all of the digested nutrients break open for other cells to pick up and use. Now the opposite of apoptosis or programmed cell death is necrosis, which is unprogrammed cell death or cell death caused by outside factors like UV light and stuff. So lysosomes play a pretty key part in that. And lysosomes are made out of the rough ER and shipped to the Golgi and then form from the trans face of the Golgi. And so going back to the apoptosis idea, it also plays into the idea of autophagy, which is the recycling of materials of certain cell components. So not the whole cell itself, so other organelles can use it. So for example, if the nucleus had a enzyme that, or okay, I guess the nucleus isn't really that good of an example. Okay, if a vesicle was transporting an enzyme that would help the nucleus make RNA faster, but then the vesicle is just like dawdling around and not doing anything, the lysosome could like consume the vesicle and release its contents to the nucleus. That's not that good of an example, but yeah. So people with Tay-Sachs disease, they have abnormal lysosomes in that they don't have an enzyme in their lysosome that can consume fats. And so eventually lipids and fats accumulate in their like nervous systems, and they can't develop properly. And so this is a really prevalent inherited disease that's been studied a lot. So here are some diagrams. So the first one is the cistern maturation model. So as you can see, the Golgi is like constantly moving in arrows back and forth. The lysosome, here's what it looks like, and the whole endomembrane system in its entirety. Okay, so vacuoles are another part of the endomembrane system. And this is because one day scientists decided to assign a fancy name to a big vesicle. So basically, vacuoles are huge vesicles that are membrane bound. So they're derived from the ER in Golgi. So because their membranes are like connected, they are part of the endomembrane system. And so because they have this compartmentalization, the inner environment of the vesicle is different from the outer environment. So there are many different types. Food vacuoles store nutrients. Contractile vacuoles, so in fresh water environments, organisms living there may have a hard time regulating the amount of water and salt that they have in their body, so they use contractile vacuoles to sort of like pump it in and out. And some vacuoles can even perform functions like lysosomes, and also they can store stuff like pigments. And so all mature plants have something called a central vacuole, which is basically a bunch of small vacuoles that congregated together. And so this central vacuole is this plant main source of inorganic ions. So also going back to last class, an organic compound has carbon in it. So inorganic ions don't have carbon in it. So potassium is an element, so is chloride, and neither have carbon. But they are still very essential to plants' growth. And they can... Okay, sorry, I blinked. So they can also absorb a lot of water, and they can actually go up to like 96% of the cell's volume. And so remember that plant cells have something called a cell wall, which is basically like a tough barrier outside of the membrane. So once they have this central vacuole, the vacuole swells up the cell and basically pushes against the cell wall, making it really tough and applying a lot of pressure. And so this pressure is called turgor pressure, and it helps plants on the whole plant scale stand up. Okay, moving on to something that is not part of the endomembrane system, mitochondria and chloroplasts. So if you are wondering why I grouped these two together, well, it's because they are both a product of something we like to call the endosymbiotic theory. The endosymbiotic theory is the idea that eukaryotic cells ate smaller bacteria, and bacteria are I was going to say powerhouse of the cell because I saw Alan's text. Bacteria are prokaryotes. So we usually associate mitochondria with ones being proteobacteria, and chloroplasts used to be cyanobacteria. And so the proof for the endosymbiotic theory, mitochondria and chloroplasts both have double membranes, as do eukaryotic cells. They have their own DNA, so they can self-replicate. They have their own ribosomes, and basically they can perform everything a cell can do alone. But they are integrated so that processes like cellular respiration and photosynthesis are more efficient. So the mitochondria is indeed the powerhouse of the cell. And so it has a double membrane, and inside there is something called a mitochondrial matrix, which is also made of cisternae, flattened sacs that go back and forth. And so within this matrix is cellular respiration happening. So cellular respiration is how we make energy for the cell, which is why it's called the powerhouse of the cell. Also, mitochondria DNA, all of it is from the mother, because the only thing you get from your dad when you are a fetus, or even smaller, when you are a zygote, a fertilized egg, is the nucleus. That's the only thing you get from your dad. So all of the DNA in mitochondria is maternal. Chloroplasts also carry out energy processes, except it basically makes its own food. So we can't do that, unfortunately. Standing out in the sun and being able to eat would be great, but only plants can do that, unfortunately. So plants use something called chloroplasts to make sugar. Chlorophyll is what absorbs the light and turns it into this food source. And it is also double membrane bound. And instead of the mitochondrial matrix, there's something called the thylakoid sacs. So thylakoids are like, wait, this will all be in the diagram next slide, so I'll wait until then. Another kind of organelle that's part of the endosymbiotic theory is just plastids in general. And that's basically just a fancy name for any double membrane organelle that has things that help certain organisms make their own food. So that can be like pigment. The chloroplast is the placid, amyloplast stores starch, chromoplast stores yellow pigment. Yeah, so here is an example of the chloroplast. As you can see, there are little stacks of thylakoids called the granum. In the mitochondria, you see the matrix that is running back and forth. And at the bottom is something detailing the endosymbiotic theory and how things went in. So yeah. Okay, peroxisomes. I promise there is not that many slides left. So, peroxisomes are not like mitochondria or anything in the endomembrane system. They're just basically an organelle bound by a single membrane, and basically hydrogen from materials in the cell combined with oxygen here to form hydrogen peroxide, hence the name. And so this basically detoxifies and breaks down a lot of things. So liver cells have a lot of peroxisomes which detoxify alcohol, and hydrogen peroxide is dangerous for our body, but we have an enzyme in peroxisomes that actually turn hydrogen peroxide into water and oxygen. So it is A-OK. And so in plants, there's a special kind of peroxisome called a glyoxysome, and it stores fat in plant seeds and can convert fat to sugar. So that's a pretty cool process. And so it's not very well studied, and we don't exactly know how it relates to other organelles and functions. And so there is not a lot about that. So open for debate. I don't know why okay. I will speed run through the next few slides. All right, cytoskeleton. This is very dense. So do you guys want to play Trust No One today?
No, because I think that you'll just terrorize us again.
Well, I can't terrorize you if it's Trust No One, because you guys
Well, like you're probably gonna be able to do something to make us all lose.
I mean, you guys are gonna inevitably all lose anyway.
Sorry, we don't trust you.
I mean, that's understandable.
No one.
Like,
we trust everyone except you.
Okay, I take it as you guys don't want to play Trust No One. Well, anyway, I'm going to speed run through these slides so we have time to play whatever game you want, Alan. not Alan, Alvin. I was looking at the chat, but Alvin was speaking. Anyway, I'm sorry, I am not in it today. Okay, anyway, so basically the cytoskeleton is a network of fibers in the cytoplasm that basically helps support the cell structure and also movement. So there are three main kinds of cytoskeleton fibers. The first one is microtubules. Basically, what you need to know about microtubules is that it is basically a hollow rod made out of the protein tubulin, and centrosomes, which play a key role in DNA replication, have form using microtubules. And also cilia and flagella, which help with cell movement. The cilia is they're like tiny hairs, and flagella is like a single thing. So if you've ever seen what a sperm looks like, it has a flagella at the end. And here is an example: cilia, flagella, and this is how it is arranged. Microtubule. Okay, second type is microfilaments. They are solid rods. They are not hollow inside, and they're made out of a protein called actin. And this cytoskeleton fiber bears tension and pulling forces. So it just basically gives cell its shape, and also it helps in our muscles. Actin and myosin are the main proteins in our muscles that help us move, and they push and pull to contract. And so that is microfilament's role. Here is an example. I really love this diagram. It's so cool. Oh no, what did I do? Okay, I made the chat full screen. All right, there we go. And finally, the third type is called intermediate filaments, and it's basically a microfilament, but it's a lot bigger and tougher. Okay, cell wall and extracellular matrix. Basically, just know that the cell wall, plants, protists, bacteria, and fungi all have cell walls, and cell walls help maintain structure. They're made out of different things. Plants have cell walls made out of cellulose, bacteria, peptidoglycan, fungi have chitin, and animals don't have cell walls, but they have something called the extracellular matrix, which is made out of proteins and other carbohydrates that help maintain structure. Again, if you want to look at all of these notes, I will post it in the Google Classroom. I'm just going through it very quick right now. As you can see, this is a plasma membrane, and on the top there is an extracellular matrix made out of fibers, helps it maintain structure. Cell junctions. Basically, just know that cell junctions are like the tape of cells and allow cells to stick together and also pass materials through one another. We don't need to go into the specifics. Okay. All right.